Hello, today we will discuss about the description, pathogenesis, and epidemiology and therapy research on Shigella.
So first of all, let's define the taxonomy of Shigella. It is under the kingdom Bacteria, under the phylum of Proteobacteria, under the class of Gramma Proteobacteria, under the order of Enterobacteriales, under the family of Enterobacteriaceae, and under the genus Shigella. Shigella are slender, non-motile, gram-negative rods. It is also fimbriate, non-sporing, and non-capsulated.
For their colony characteristics, Shigella are facultative anaerobes but grow best aerobically. They are convex, circular, smooth, and transparent colonies with intact edges reaching a diameter of about 2 millimeters in 24 hours. They best grow at a temperature of 37 degrees with a pH level of 7.4 on ordinary media. On a maconky agar, they are pale and colorless except for Shigella sunei, which is a late lactose fermenter. All strains of Shigella species ferment glucose. Again, with the exception of Shigella sunei, they do not ferment lactose. The inability of lactose fermentation distinguishes Shigella on differential media from other lactose fermenting members of the Enterobacteriaceae. Example of differential media are the eosin methylene blue agar and the maconky agar. Deoxycholate citrate agar or DCA and Salmonella and Shigella agar or SS agar medium are useful selective media for the Shigella, while Wilson and Blair medium inhibits their growth. On acylous lysine deoxycholate agar or XLD agar, Shigella produces pink colonies without black centers. Other media includes is hectoin and enteric agar, which is a selective media. In metabolism, as said earlier, they are facultative anaerobic parasites, and these Shigella pathogens use a mixed acid fermentation pathway to metabolize substrates. Products of this anaerobic pathway includes ethanol, acetic acid, lactic acid, succinic acid, formic acid, and carbon dioxide. In terms of the nutrient needs for growth in culture, according to a study of Ahmed, methionine, nicotinic acid, and tryptophan showed a rapid growth in the high cell yields to oxytropic isolates of Shigella.
So the next one is the niche of the Shigella. So it is commonly found in the gastrointestinal tract of humans, as humans is the primary reservoir of Shigella. As previously described, Shigella infections are almost always limited to the gastrointestinal tract, while bloodstream invasion is quite rare. Shigella are highly communicable and can be transmitted via the fecal-oral route. The transmission of Shigella can involve direct person-to-person or sexual contact, or indirectly through contaminated food, water, or fomite, and even on surfaces that have been touched by infected people.
Now for the disease description of Shigella. The infection caused by Shigella is called shigellosis, which causes a bloody or non-bloody diarrhea to those infected persons, and also they are contagious. In the United States, there are about 500,000 cases of shigellosis every year, and up to 15% of these cases may be related to international travel. However, in comparison, there's an estimate of 90 million cases of shigellosis worldwide. Take note that most cases of this infection occur in children younger than 10 years of age. The mortality rate of shigellosis accumulates to 600 deaths per year worldwide, and 6,900 to 30,000 of these deaths are attributed to foodborne transmission. So this is a timeline after getting the infection. After a short incubation period of 1 to 4 days, there is a sudden onset of abdominal pain, fever, and watery diarrhea. A day or so later, as the infection involves the ileum and colon, the number of stools increases. Each bowel movement is accompanied by straining and tenesmus rectal spasm with resulting lower abdominal pain. In more than half of adult cases, fever and diarrhea subside spontaneously within 2 to 5 days. However, in children and elderly adults, loss of water and electrolytes may lead to dehydration, acidosis, and even death.
So the next one is the distribution of Shigella. Although 90 million cases occur worldwide, Shigella is endemic to temperate and tropical climates. Particular species of Shigella are found in different places. For example, Shigella sonnei is commonly found in industrialized countries, while Shigella flexneri is the predominant cause of Shigella in developing countries. Other types of Shigella, such as Shigella dysentery, can spread widely and is common cause of epidemic dysentery with an associated high morbidity and mortality, particularly also in developing countries.
For detecting Shigella, for specimen, fecal specimens should be collected during the early stages of the illness. Specimens include fresh stool, mucus flecks, and rectal swabs for culture, while whole stool is usually the preferred specimen for laboratory workup of diarrhea. Rectal swabs with visible fecal staining present may be the preferred specimen for the isolation of Shigella. For culture, materials are strict on differential media and on selective media. Colorless colonies are inoculated into a TSI agar slant. Non-motile organisms that fail to produce hydrogen sulfide that produce acids but not gas in the butt and an alkaline slant in TSI agar medium should be subjected to slight agglutination by specific Shigella antisera. For serology is not used to diagnose Shigella infection. Lastly, for nucleic acid amplification tests, there are several commercial NATs that directly detect Shigella in fecal samples along with some of the other major enteric pathogens. There are other molecular methods used for Shigella, and one of it is a PCR test, which is a technique used to amplify small segments of the DNA.
So the next slide is about the treatment of Shigella through antibiotics. Antibiotic treatment is recommended for the treatment of severe infections and to prevent secondary spread among people living in closed quarters such as family members or during outbreak. Ciprofloxacin and ceftriaxone are effective antibiotics of choice. However, in recent years, the Centers of Disease and Control and Prevention have identified emerging strains of Shigella species with potentially reduced susceptibility to ciprofloxacin, while ceftriaxone is commonly used for treatment of children with shigellosis. And finally, azithromycin has been shown as a useful antibiotic for treatment of antibiotic-resistant Shigella infections in adults and children.
In disease management, because humans are the main recognized host of pathogenic Shigella, control efforts must be directed at eliminating the organisms from this reservoir by number 1. Sanitary control of water, food and milk, sewage disposal, and fly control. Number 2. Isolation of patients and disinfection of excreta. Number 3. Detection of subclinical cases and carriers, particularly food handlers. And number 4. Antibiotic treatment of infected individuals.
So, to conclude our presentation, we found a research connected to Shigella entitled Manipulation of Epithelial Cell Death Pathways by Shigella, and it was conducted last year. It shows how Shigella manipulates cell death in order to infect more. First of all, there are three means of regulated cell death in response to infection. First is pyroptosis, apoptosis, and necroptosis. In this study, they showed that Shigella uses the so-called T3SS effector OSPC3 to prevent pyroptotic death, and the so-called OSPD3 in Shigella acts as an inhibitor of necroptosis. In conclusion, by having OSPC1, C3, and D3, Shigella delays the epithelial deaths long enough to permit replication to high levels, followed by an invasion of neighboring cells. With this delay, there is ultimately more catastrophic epithelial damage and inflammation with the release of large numbers of bacteria into fecal matter. And as we all know, fecal matter is one way of transmission of Shigella.